I have to say that I've been impressed by the number of people that came to the symposium and how far they'd be willing to travel. I know that people came from, Berkeley doesn't count, UCSF, and, a little bit further in Stanford, Davis, Merced. But our next speaker come from Germany, so I'm not sure the only reason was the meeting. So he's a postdoc, and he will be talking today about, influenza and export from the nucleus.
All right, so thank you for the introduction. I'd also like to thank the organizers of the symposium for giving me the chance to talk today. And I promise you I will keep this short so that you don't have to wait too long for lunch. So as you can see from the title of my talk, we're also interested in the interaction between virus and host cells. In this case, it's the influenza virus. And as I'm sure all of you know, the influenza virus is a respiratory virus. It causes both epidemics and recurring pandemics in humans. And influenza virus is a negative strand RNA virus. And what this means is that the genome of influenza virus always exists in the form of the viral ribonucleoprotein complex, or vRNP. That's going to be pretty important for our talk, so let's go quickly through the vRNP. It consists of a single-stranded RNA molecule, which is encapsulated by the viral nucleoprotein NP. And it's associated with the viral RNA polymerase, which in the case of influenza is a heterotrimer consisting of the PA, PB1, and PB2 subunits. Now, vRNPs perform quite a number of enzymatic functions in an infected cell. And one of the main questions of our lab for quite a number of years now has been, can we identify either cellular or also viral proteins which associate with vRNPs and help modulate all these different functions that one protein complex carries out? But now in the case of influenza, this question is a little bit spicier because influenza carries out all of these functions in the nucleus of the infected cell, which means that it also has to at some point import the vRNPs early in infection into the nucleus, and then at a late time point of infection, export the newly assembled vRNPs back to the cytoplasm so that they can bud. And the nuclear replication cycle of influenza also makes life a little spicier for the researcher because the nucleus is a very difficult organelle to work with biochemically. So to kind of subvert some of these problems, our lab has developed two tools over the last few years to study nuclear vRNPs. First tool is a recombinant influenza virus, which expresses an affinity-tagged vRNP. So the way this works is we take the lab strain of influenza called WSN, and we've genetically fused a strep tag to the C terminus of the PB2 subunit of the viral polymerase complex. So using this virus, we can infect cell cultures and then purify large amounts of vRNPs directly from the cells, or the lysates. The difficulty, though, is even with this virus, I said there's a large number of vRNPs performing different functions more or less simultaneously in the cell, which means that even when we purify the vRNPs, we still have a very crude mixture that we can't really analyze very well. So to try to biochemically separate the different functional species of vRNPs, before we purify them from infected cells, we first fractionate the nuclei of infected cells into different biochemical fractions. Now, this is an adaptation of a fairly standard subnuclear fractionation scheme. And what this leaves us with are three nuclear fractions, one of which contains most of the soluble nuclear proteins, which we call the nucleoplasm. Then we have two chromatin fractions, one of which is enriched for markers of transcriptionally active chromatin, which I'll call here somewhat unscientifically open chromatin due to sort of open nature. And then the other chromatin fraction is enriched in more transcriptionally inactive chromatin, which we call the dense chromatin fraction. And as it turns out, if we perform this subnuclear fractionation before we purify the RNPs, we can actually obtain fairly homogeneous populations of RNPs performing these different functions. Now, the function I'm going to talk about for the rest of the talk today is the export of vRNPs, which occurs during late time points of infection. And the reason I'm really interested in the vRNP export is because for me, it's sort of the paradigm for the really intricate dance you have between the virus and the host cell. So what was known about the export of influenza vRNPs when we started was known as dependent on the cellular protein Crm1, which I'm sure many of you are familiar with. And it was also postulated to be dependent on several viral factors. These include the viral matrix protein, which is called M1, also the viral nuclear export protein, hence its name, which is also known as NEP. And when we started, the hypothetical model of how this export would take place was simply that all of these factors would latch onto the vRNP, and that this complex would then be exported to the cytoplasm. However, there are two slight problems. This complex had never actually been demonstrated in any physical form, either in vitro or from cell culture. And there's quite a bit of controversy about whether these different factors were in fact necessary. So the question we asked is, could we use these two tools that I just described to you to purify this type of complex from infected cells? And so the strategy we took to try to purify this complex was we took advantage of the fact that there's a chemical inhibitor of Crm1 known as leptomycin B. And if you treat influenza-infected cells with leptomycin B, at a late time point of infection, the vRNPs become trapped inside the nucleus. So the way we carried out the experiment, essentially we infect cell cultures with this recombinant virus I was just showing you, in either the presence or absence of leptomycin B. And then at a late time point of infection, we harvest the cells, fractionate the nuclei into the lysates I showed you, and from each of these fractions, we then purify the vRNPs using affinity purification. And now what we postulated is, if in fact we've been able to purify these export complexes, then we should have one of these nuclear fractions where the amount of RNPs that we purify is much higher in the presence of leptomycin B than in the absence of it. And furthermore, those RNPs should also be associated with either M1 and possibly also NEP. OK, so let's look at the data here. So now this is a Western blot just showing you the vRNPs that we purified from the different fractions of the nuclei. So over here on the left, these are cytoplasmic RNPs. And then here we have those three fractions I was telling you about. In this case, we're looking at PA as a marker for the polymerase complex, and then NP, the nucleoprotein, both main components of the purified vRNPs. And as you can see, so in the left column of each of these, we have the yield of vRNPs without leptomycin B. And on the right, we have the yield in the presence of leptomycin B. And as you can see, we do, in fact, have a fraction where there's a strong enrichment of vRNPs after leptomycin B treatment, namely this dense chromatin fraction. And if we now look at these same purified complexes to see if either M1 or NEP is associated with them, what we see is, in fact, in this dense chromatin fraction, both proteins are associated with the complex. And together with some other data that I don't have time to show you today, we're very confident that these fractions that you see here, or this fraction you see in the right-hand column, represents this export complex that has been sort of searched by several groups for many years. So that in and of itself, OK, kind of nice. What was really surprising, though, first with this result, though, or I should say also very interesting, was the fraction that we were able to purify this from, namely this dense chromatin fraction. And the reason that was interesting to us is because, first of all, in order to solubilize this fraction, we had to treat the nuclei with 500 millimolar of salt, which means that these complexes were very tightly bound to the chromatin. And that's surprising because what's known about export complexes that use Crm1 is that they're actually very mobile, very dynamic complexes in the nucleus. And the second reason is because we took a look at several cellular proteins that are known to be exported out of the nucleus using Crm1, and none of them ever show up here in this dense chromatin fraction, which makes sense based on their function. So we thought maybe influenza is using some kind of unique mechanism to export its vRNP complex. And to dig further into this, we asked ourselves, well, where's Crm1 in the nucleus when all of this takes place? So in order to look at Crm1, we just took cells here. This isn't purified. This is just looking at the lysates of cells, just at the different fractions within the nucleus, trying to figure out where do we find Crm1. And as you can see here in uninfected cells, you find most of it in the soluble part of the nucleus or associated with transcription of active chromatin. This fits with the literature very well. But what you see is when we infect with influenza virus, there's a distinct population of Crm1 that's now accumulating on this dense chromatin fraction where it otherwise isn't found during a normal cell function. And so what this suggests to us is that during influenza virus infection, these vRNPs, when they're assembling their complex directly before they're exported, they sort of park themselves on this transcriptionally inactive chromatin and possibly somehow recruit the cellular protein Crm1 to that site. And now to confirm that the Crm1 is actually located at the same site where these export complexes are being assembled, we performed immunofluorescence. But in this case, the immunofluorescence isn't of normal cells, but rather we also fractionated the cells so that what you're looking at here is the dense chromatin fraction of the cell. So what you can see here is when we infect cells with influenza virus, Crm1 does indeed accumulate on this chromatin fraction, and it co-localizes very well with the viral nucleoprotein, which is a marker for the vRNPs. And this is only seen at very late time points of infection when these export complexes are being formed. So what this suggests to us is that indeed the virus is recruiting the cellular factor to this domain on the chromatin, which it normally would never be found in. And what we asked ourselves is, does this recruitment possibly mean that it's depleting the pool of Crm1 available for other proteins in the nucleus. And to measure this, we took a marker for Crm1 export, a GFP, with a nuclear export sequence from a Rev fused to it. And as you can see here, at a late time point of infection, there is in fact a significantly more greater amount of GFP found in the nucleus of infected cells than uninfected cells, which suggests that non-viral Crm1 substrates are exported less well from the nucleus when vRNPs are being exported. And we also looked at this data statistically, and as you can see, this only happens at a late time point of infection when vRNPs are being exported. And now, we have data that I don't have time to show you, but what we found out is that this phenomenon that you see here is not actually due to the export of the vRNPs, but is actually due to their targeting to this inactive chromatin that I keep talking about. So it's the location of the vRNPs on this chromatin that delays or sort of retards the export of other Crm1 substrates in the cells. So just briefly, the last question that was open to us is, why is this inactive chromatin or whatever dense chromatin, how does targeting the vRNPs here confer an export advantage to the virus? And to solve this, we went back to the literature to find out, does chromatin play any role in nuclear export? And as it turns out, it does. And the way that is that Crm1 export can only function when Crm1 is in complex with the small GTPase RAN, and that can only function when RAN is complexed with GTP and not GDP. And like all GTPases, RAN has a cognate guanine exchange factor. And this is a protein known as RCC1, and as it turns out, RCC1 is located on the chromatin. And if we take a look at which part of the chromatin, the open or dense chromatin, we find more at, it turns out it's most highly enriched on the dense chromatin fraction. So we asked ourselves a very basic question. If we have our VNP export complexes in this dense chromatin and the protein which is responsible for essentially driving the entire gradient of nuclear export in the same fraction, is it possible that they're also physically associated with each other? And I'll just show you one brief slide here. We performed a simple experiment. We took the chromatin of infected cells and immunoprecipitated RCC1. And in fact, we could co-immunoprecipitate PA, NP, and M1. And NP we weren't able to detect. But based on everything we know about the nature of vRNPs in the cells, the fact that we were able to co-immunoprecipitate these three proteins suggests that this is, in fact, a vRNP export complex, which is physically associated with RCC1. So based on these data, we've postulated a model of how export might work both in uninfected and in infected cells. So the model we have is that in uninfected cells, like I said, you have these complexes containing both Crm1 and RAN GDP, and these need to be converted to RAN GTP in order for export to occur. This occurs on the chromatin. However, all the data suggests that the cellular proteins which are exported by Crm1 are not located on this transcriptionally inactive chromatin, but in other domains of the nucleus. And therefore, there has to be a movement to a distal site. And then our hypothesis is that at a late time point of influenza virus infection, the virus assembles this export complex on the chromatin directly next to, or directly in physical proximity or contact with RCC1, and is thus in a perfect location to essentially snatch these newly generated cellular export machinery complexes before they can be used by the cell. And in other words, it can ensure that its complexes, or that its proteins get exported in preference to cellular substrates of Crm1. Okay, so with that, I'm at the end of my talk. I'd like to thank all the people who helped me out with this project, including my PhD advisor at the University of Freiburg in Germany, Professor Martin Schwemder. I'd also like to thank our collaboration partners, especially Nadia Nafak at the Institut Pasteur in Paris, and Thorsten Wolf at the Robert Koch Institute. And I'd also thank all of you for delaying your hunger another 15 minutes for me.
I have a question, actually. I've seen your nuclear export protein. It doesn't seem, when you do UIP, it doesn't seem to be as abundant.
I'm sorry?
Are you sure about the nuclear export proteins? Right? Mm-hmm. It doesn't seem to be as abundant when you complex. So are you sure about the stoichiometry, or do you have like two different complexes?
Our speculation is that there's only a very small amount of NEP which is actually required for the export to occur, perhaps as little as 1 molecule per complex. And that's based on the fact that if you create a virus which expresses only about 2% of the normal NEP, the export still occurs very well. So that's our postulation, at least.
Martha?
Or, oh yeah, just say it now.
So Crm1 also affects the export of some RNAs. Do you know if RNA export is at all affected by the influenza infection?
I'm sorry, you're saying, do I know?
The CRM1 pathway also affects the export of at least some of RNAs, although it's not the major pathway. Do you know if RNA export is affected by the influenza infection?
We didn't look at RNA export. On the other hand, influenza virus, or during influenza virus infection, there's very little cellular RNA that's produced anyhow, or mRNA that's produced anyhow, because the virus snatches the caps of the pre-mRNAs. So we didn't look at that for that reason. I'm sorry, I didn't hear the.
Why would the RCC?
I think that's just for the.
Why would the RCC1 being in the dense? What is the advantage for the RCC1 being in the dense chromatin fraction when it's actually mainly needed in the open chromatin fraction? When the CRM1, not in the infection, in the non-infected state.
That's a very good question. The model for CRM1 is it is essentially always scanning the nucleus back and forth. That it's an extremely mobile protein, so presumably it's mobile enough that it would always be coming into contact. And even though biochemically this dense and open chromatin are separate fractions, physically they're most likely very close by to each other. So that a mobile protein like CRM1 would still have access to RCC1 even though it's biochemically on a separate fraction than CRM1. But that's pure speculation on my part. RCC1, yes, it's actually RCC1 is regulator of chromosome condensation. So during cell division, it's latched on to the chromosomes all over the place.
Okay, thank you.
All right, thank you.